Okay, so today we're going to talk about photosynthesis. And again, I put all of the sorry, put all of the essential knowledge. Oh my goodness. Okay, here it is. Okay. I've never had this happen to me before. Oh, here we go. Okay. So as you can notice, there are a lot more than there usually are here. But a lot of them are repeats. So, like everything here is basically about the input of energy and the laws of thermodynamics. And then these are the, I guess I would call them standards, or the knowledge that actually relates to photosynthesis. So, what does this picture have to do with photosynthesis? Yes, there's green plants, and the green plants are receiving a a variety of wavelengths of light from the sky. And the green plants will use some of those wavelengths and not others. So it can use some more efficiently than others, and that is why this beautiful picture is here. Do you care to see the video? Okay, typically we think of plants as the main producers or the main autotrophs in our ecosystems, but in addition to plants, there are different types of algae that can photosynthesize. And if you look and see here, there are unicellular protists like Euglena. They contain chloroplasts. Cyanobacteria used to actually be classified as a plant until recently when DNA analysis allowed us to realize that it's actually a bacteria, not a plant. And then purple sulfur bacteria, they are also autotrophs, although I'm not convinced of their use of photosynthesis. But it would be really interesting, and it is a really interesting field of study currently, where we can harvest the energy from the sun and turn that into fuel that we can use. And so people talk about alternative fuels or fossil fuels, but the interesting thing is the fossil fuels come from the plants. They have decayed and been compressed, or basically, but they come from plants. But all of the, or many of the alternative fuels that we're looking at also use plants as a source of energy, just living plants rather than dead ones. So chloroplasts are the organelle in which photosynthesis actually occurs, and leaves are the main site of photosynthesis in any plant. And chlorophyll is the pigment that gives the leaves their green color, and the chlorophyll is found within the chloroplasts. So chloroplasts are found in the mesophyll, or the interior tissue of the leaf. The prefix meso means middle. So the mesophyll is the interior tissue. And each mesophyll will have 30 to 40 chloroplasts approximately. But that's going to depend on quite a bit. So carbon dioxide will enter and oxygen will exit the leaf through microscopic pores called the stomata. Remember, we've also heard those referred to as stoma or stomates. And the chlorophyll is actually present in the membranes of the thylakoids. Those are those pancake-looking things, and you stack them together, and they're called a grana. And then surrounding those in the chloroplast is a fluid called the stroma, and that's sort of analogous to the cytoplasm in a cell. So here, if you look, here's the cross-section of the leaf. You can see in the mesophyll, oh yeah, I forgot I can write. In the mesophyll, hey, why didn't it let me write? It did the other day. Well, that's sad. Well, over here in the mesophyll, you can see that there are lots of chloroplasts, and it shows you where the stomata are located. And then this portion here, this is the entire chloroplast. And if you were to look at it with an electron microscope, this is what you would see. And each of these dark stacks are the granum. So photosynthesis Photosynthesis is again an imprecise formula, but we can summarize it using this formula here. 6CO2 plus 12H2O plus light energy yields C6H12O6 or glucose plus 6O2 plus 6H2O. So our reactants are going to be the carbon dioxide and the water, and the chloroplasts they split the water into hydrogen and oxygen, and then as you can see, all of the carbon and some of the oxygen is used to make the sugar, and some of it is used to produce more water. And then we take the hydrogens from the water and we use that to make our sugar and the water product, and also that's where the oxygen gets released. Of course, this is a redox reaction or a redox process, so it's going to reverse the direction of electron flow when compared to respiration. So those two processes are coupled. Photosynthesis is a redox process, and water is oxidized and the carbon dioxide is reduced. It's an endergonic process, and the energy that it needs is provided by sunlight. So this is showing you how the energy plus the carbon dioxide gets reduced to glucose, and then the water gets oxidized to oxygen. So there are two main stages of photosynthesis. We have the light reactions, which require light energy, which is the photo part of photosynthesis, and the Kelvin cycle, which is the synthesis or the building part. And sometimes those are also called the light-independent reactions because light isn't required for those reactions to take place. So the light reactions are going to convert solar energy to the chemical energy of ATP and NADPH, and that will be used to create sugars. So the light reactions are going to occur in the thylakoids. The first thing that will happen is that water is split, the oxygen is released. We saw this in the video. Do you recall? So NADP plus becomes NADPH, and ATP is generated. Then the products there go through the Kelvin cycle, which occurs in the stroma. So the CO2 will form sugar using ATP and NADPH. Then the Kelvin cycle starts with what we call carbon fixation, and that incorporates carbon dioxide into organic molecules. So this should look like a familiar picture to you. I'm glad it does to some of you. But we see this picture sometimes on your Regents exam. So basically it just shows you that in the light reactions it requires light, water is split, and oxygen is released. And then you've got these carrier molecules that go to the Kelvin cycle where carbon dioxide is split, giving us some sugars. Sunlight is a form of electromagnetic energy, sometimes referred to as electromagnetic radiation, and like many other types of electromagnetic energy, light travels in waves, and the wavelength, if you recall, is the distance between the crests of the waves, and that's going to determine the type of electromagnetic energy. Have you guys learned about wavelength? Shall I give you a mini tutorial on wavelength? No? Okay. So all of the range of electromagnetic radiation that's out there is referred to as the electromagnetic spectrum. And we use a lot of visible light for everything because that's what we can see by. So visible light consist of the wavelengths that drive photosynthesis. Okay, and those are the colors that we can actually see. So light behaves not only as waves, but it also behaves as particles, and we call those particles photons. So that's important to remember here as we learn about the next step, that light behaves as both waves and particles. So at the top here, this bar or this graph represents the entire electromagnetic spectrum, and as you can see, visible light is a very tiny portion of the entire electromagnetic spectrum. So, but visible light are nanometers from approximately 380 to 750 nanometers. As you can see, the shorter the wavelength, the higher the energy it has, and the longer the wavelength, the lower the energy. Okay. So we have to have some kind of substance that's able to absorb this light or absorb these wavelengths, and those are pigments. Pigments are defined as substances that can absorb visible light. And different pigments are going to absorb different wavelengths of light, and wavelengths that are not absorbed will be reflected back or transmitted through the leaf. And leaves appear green because chlorophyll is going to reflect and transmit green light, like so. Okay, we can use a spectrophotometer to measure a pigment's ability to absorb various wavelengths. However, we don't have one, so we use a colorimeter. So a spec is a little bit different than a colorimeter in that, you know how when we use our colorimeter we have to choose a particular wavelength that we want to look at? The spec will allow you to set it to any wavelength that you're looking at. It could be just any wavelength, and it will simply measure the transmittance at each one of those wavelengths that you set it at. So it's slightly different. It's more, it allows you to collect a broader range of data when you're using a spec. So here is a picture of one. Do you need me to go back? Oh, what does it say on your page? Oh, it just, oh. Okay. Are we okay? Okay. So this is showing you if you were to take white light and use a prism to refract the light and separate it into its color components, and then put it through a filter that allows only one color of light to go through. Right here on the top one, green light is being shown into a chlorophyll solution, and then what is transmitted is measured by a photoelectric tube. So this tells you that 100% of the light that hits the phototube was transmitted. Therefore, very little of it was absorbed. Does that make sense? So on the other side, if you did the same thing, only this time you allowed blue light through, and blue light hit the chloroplast solution, and then what was transmitted went to this photoelectric tube, it shows you a very low transmittance, almost zero, which means that blue light is absorbed by the chlorophyll solution. So we can take this data and make an absorption spectrum, which is a graph that plots the pigment's light absorption versus its wavelength. And the absorption spectrum of chlorophyll A, because there are two types of chlorophyll mainly that we look at. So chlorophyll A suggests that violet, blue, and red light work best for photosynthesis. But we can also look at an action spectrum, and the action spectrum profiles the relative effectiveness